Hello, welcome to your own channel, Curious Corners in Biology, which is a channel of delving deep into this subject, discussing the scientific aspects of biology, and thinking about the curiosity in this subject. So, if you are new to this channel, I hope you have understood the description of the channel. Here we discuss inquisitive, curious questions in biology, and you can check out my book in the description below, in the link below, Curious Corners in Biology, where you will find many questions in different topics of biology which require scientific inquiry. Okay, so today's topic of discussion is we know that animal cells have centrioles, but then plant cells do not have them. Why so? Okay. So, first of all, plant cells have thought to have diverged from a common ancestor from which animals and protists have evolved. So, the common ancestor of plants may be different, or the ancestor of the plant may be different from protists and animals. Protists and animals do have centrioles, but plants do not. So the question is, what replaces the function of centrioles in plants? The answer to this is polar caps, also called as spindle pole bodies, SPBs. What are these? These are simply called as MTOCs, microtubule organizing centers. These are sites, or you can say places, from where the polymerization of microtubules will start, from where spindle fibers start to form. Okay. But in animals, we have specialized organelles which are called as centrioles, and they also do the same function. Now, in plants, the evolution of these cells is different. Plant cells. Plant cells have evolved a method of mitosis called as endomitosis, where nuclear membrane does not break down, remains intact during the entire process of mitosis. And spindle fiber, if it forms within nuclear membrane. So here centrioles are not required within the nuclear membrane. So the reproductive strategy which plant has evolved is different. Okay. And there is one more point to be taken into consideration, that we know something called as endosymbiotic theory in biology. Endosymbiotic theory states that prokaryotic cells like bacteria, cyanobacteria, were thought to have been integrated in eukaryotic cells, must have developed a symbiotic relationship, and then became chloroplast and mitochondria of eukaryotic cells. Cyanobacteria became chloroplast, and bacteria, eubacteria, became mitochondria in eukaryotic cells. So, in this process, it has been thought that the prokaryotic cells, so evolved in chloroplast and mitochondria, might contribute into a regulation mechanism where eukaryotic cells like plant cells would not require centrioles. Now, what is the exact mechanism? How is this expressed through genes? What are the kind of genetic regulations involved? Is again a topic of research, but this is one hypothesis which also comes into picture. Okay, so these are various reasons why we don't see centrioles in plant cells. The next curious question which I would like to discuss is why centrioles are not membrane bound. Okay, we know that there are three types of organelles in eukaryotic cell: those which are membrane bound, those which are double membrane bound, and those which are membraneless. Examples of membraneless organelles would be ribosomes and centrioles. Now, centrioles, as I mentioned, are also called as MTOCs, microtubule organizing centers. They do not require a membrane because they would directly like to polymerize microtubules or spindle fibers, which are mostly supposed to attach with the chromosomes to facilitate chromosome movement during cell division, like mitosis and meiosis. membrane development is of no use. It is dysfunctional. Okay. So there is no need for evolution of membrane. That is why we don't see any membranes. The other question along the same line is why also ribosomes are membraneless. This is because ribosomes has a function of protein synthesis. Now, as soon as protein is synthesized, it doesn't need to be packaged within the membrane because all the chemical modifications would anyway take place in cytoplasm. And of course, ribosomes through translation carry out protein synthesis by associating with mRNA and tRNA. And mRNA and tRNA are also directly present in cytoplasm. So actually there is no requirement for a membrane even in case of ribosomes. Okay. Then just as we have discussed in one of the previous videos that how does cancer affect mitochondria, a question arises how does cancer affect ribosomes in a cell? Okay. So ribosomes, as we know, are involved in protein biogenesis or protein synthesis. Straight away, what happens during oncogenesis, that is formation of neoplasm or onset of cancer, is that ribosomes simply proliferate in number. That means the amount of protein synthesis would be very high. What causes this? It has been believed that there are certain oncoproteins which are expressed by oncogenes. These oncoproteins will simply cause proliferation of ribosomes, and ribosomes will start to synthesize more and more protein. So such proteins are unwanted, and we see a heavy load of proteins, ribosomal proteins, in cancer cells. So one thing which can be targeted during cancer therapy is ribosomes itself, because the amount of ribosomes are increasing in cancer. So ribosomes become target sites for cancer drugs. And of course, many drugs are devised based on this approach as well today. And yes, much of the research is going on. So, this is what I would like to discuss today. If you are interested in such curious questions in biology, of course, in the coming days, we are also going to discuss something more except the topic of cells. We are going to discuss n number of topics and n number of questions. Okay. So, the ultimate motto of this channel is to make this subject more interesting. Okay. And to discourage rote learning, because there is a common perception that biology is rote learning and it's a descriptive subject and stuff. So I just want to uproot this concept. Okay. So check out the book. The book is very interesting. Curious Corners in Biology. It's available at a very affordable price. Okay. Just the cost of your pizza. Okay. So we'll see you next time. Thank you. Bye.